Okay, last day of the class. We will talk about virus, because in the last 4 years, you will heard about the virus almost every day. There's a period of time. So we talk at the end, still give you some information about the virus, roughly about 30 minutes. Then we're going to go over the review exam of exam 4. Okay, so let's talk about virus. This is an overview picture of the virus. So we can draw what a virus looks like. Virus, you need to know, this is acellular. Acellular, which means cell is not a single unit. It is an obligate parasite. What this means, they need a host. They will not survive just temporary. They will not be survived without a host. So there is a period of time. When the beginning of the COVID-19 in 2020, people say they shipping from the Amazon. Okay, there is a box. The virus from the box will be transferred. Then everybody is going to do the sanitation of those things. So what do you think of that? It may be survive on the box surfaces, but the time is very short. They survive on the box of surfaces, not because they can survive on. The reason is on the box surfaces. There are some of the material there can let them survive like a host. So possibly, let's say there's a bacteria there, and they could be survived there. It's like a fudge. Okay, so just want to let you know, and more or less, they need the host. So what it looks like, the structure is very unique. So we can draw a structure. Before we do, we just draw like this. Like that. The outside is protein. This inside is nucleic acid. And the nucleic acids could be DNA or RNA. This is all, not and, because never both. This is the key. And then you're going to see these things like that comes out. We call it spike. These could be invaded into the host. And the procedure is very similar to what we talked about before about the fungi. We can say it's an ovoid body if they invaded into the host. And these spike, if they looks like a crown, then we talk about the coronavirus. And the coronavirus in 2019, they gave you, that's what happened. And those spikes could be moving. They will generate lots of the mutations. That's why you have like Delta, you have like Omicron, all those type of things happens. But the idea is, when they transferred from human to human, they will become weak and weak and weak. Obviously, with a huge population, the immune ability will also increase. So that's something happened. So these days we're still existing, but nobody cares about it anymore because the toxication is relatively weak compared to when it started in 2020. Okay, so this is a big picture of what we talk about. Here, we have a couple of the 5 dots talk about virus. It's an obligate parasite. It cannot do independent metabolism, need a host. It's relatively small, but something could be large. Let's say polio, it's only 20 nanometer. The pox virus could be 400 nanometer. So, filterable, what this means, if it's a bacteria, they can be filtered if you're using 0.22 micrometer filter. But a virus will be passing it. So pass through membranes. Last one possessing one single type of nucleic acids we mentioned is either DNA or RNA and never both. Classification of the virus, so it depends on what type of the nucleic acids. It could be single-stranded, double-stranded. And we also talk about this protein, we call it capsid. And based on the capsid morphology, we could name a different type of the virus. Hexagonic, endogonic, all kinds of things. Envelope, we will say it's a naked virus, enveloped or enveloped virus. Host range, we could say the animal virus, human virus, and based on the host. And this is I already drawn, the structure of the virus, and you can see what it looks like. Capsid nucleic acids, a non-enveloped virus. Enveloped virus is a little bit complicated. You have a spike, capsid, nucleic acids, and envelope. Now, this is something you need to know, because a virus structure is relatively simple. You had a protein, you had a nucleic acids. So, there are not many of the targets can work for the drugs. That's why the antiviral drugs very limited from the history standpoint. This is the reason when you get a coronavirus, there is never have any medicine. The physician will give it to you, said, okay, drink this bottle for 3 days and you're going to be recovered, because it's very difficult to focus on the targets to kill the virus. If we develop some of the medicine, what will happen? The target will be protein. Then the human being has a protein, animal has a protein. So we'll have a side effects. Sometimes it's very strong. And if they are working for the targets is nucleic acids, then obviously we already learned in the exam 3, there are lots of the mutation will generate and a lot of the bypass for the virus to survive during the replication, transcription, and translation. So that's a reason in the longer time there are not many very effective antivirus drugs. And they developed a couple during the COVID-19 time, but it's not very, some of them are not very effective. The best thing we can say to control the virus is improve the immune ability with the help of the vaccination. So that is what the textbook talk about that. And you also need to know when the non-enveloped virus compared to the enveloped virus, it is very surprisingly, enveloped virus is easily to be destroyed by drugs because there are more targets they can work on, especially the envelope. The envelope, you can think about, it's a quotation mark of bacterial cell wall. Same thing like a cell wall of a bacteria. It's just for the virus. And this tells you you have a different type of the virus. You have a vaccinal virus, herpes virus, rotavirus, phage, bacterial phage, and tobacco mosaic virus. That's most studied. And the minivirus, human polyovirus, so all kinds of the more different morphology. So they talk about the capsids. The capsids is basically protein. And the protein have their very single units. That single units is called protometer, a protomer. Sorry. it's called protomer. That's a single unit composed to capsid, and the major component is protein. And there are lots of the subunits, and the morphology is very different. So it could be helical, icosahedral, or complexed, which means a multiple different morphology combined together. So this is an example. This is a helical capsid, which is a tobacco mosaic virus, because it's huge. So they studied it very well, the structure. And the inside you see the RNA. So we can say this is the RNA virus. And outside you see those protein composed by protomer. The protomer is very organized, looks like a flower. It's like a skirt, and it's very, very long. And people saying that the size of the RNA is almost equal to the size of the protomer. And the size of the capsid is a function of nucleic acids. And we have some other examples. Icosahedral capsids. So you can see that capsomer is made by different units. If it's made by 5 subunits, we call it pentons. If it's made by 6 subunits, we call it hexamers. So this is using electron microscope can see the morphology is very clearly inside. And that is very interesting because all these morphology and structure sometimes relate to the pathogenic effects of this virus. Then based on that, they will find some of the targets, they could develop the drugs, or sometimes they could be even isolated, then we'll see develop the vaccines. So that's an interesting talk about the structured biology. So this is the one, is a complex capsid. And this is a typical example of a bacterial phage. We will mention this real quick, we mentioned it briefly. So you see inside the nucleic acids, capsid head, they have a collar, teeth, the base, and the tail fibers. Then they're using an electron microscope, then cut the cross section, then you see the picture showing looks like that. And they have a longer tail, and they have a base plate, and the pins, that area is easier for them to be attached to the bacterial surface, and then the nucleic acids could be invaded and could be cause lysis of the bacterial cell, or they could be integrated into the bacterial cell, then become a temperate phage. The genome structure we just mentioned, it could be double-strand DNA, single-strand DNA, double-strand RNA, single-strand RNA, and the chromosome size is varies. Some of them are very small, 4 or several hundred gene base, but some of them could be a segmented genome, which means only as small as 8 pieces of RNA. For example, influenza virus. This is part of the reason is influenza is difficult to control to develop the drugs, because RNA pieces is relatively so small. Some of them is only like 2. If the mutations, that happens only like 4 or 6. It's a limited amount, so limited area for the targets of the drugs. Now, this is easy to understanding is that when the virus invaded into the host, they always have an eclipse phase. It's kind of like the viral infection that disintegrates, and you're not going to test the infectious particles in the cell at this time. But the body started to show some of the symptoms, but later on, with the development of the infectious, they will be reappear in the cell. The virus we mentioned, it is an obligate parasite, so they need a host. And the host is lots of the times is very specific. So for example, the phage, that is specific for bacterial host. And the HIV infector will need the cells to display cluster differentiate 4, which means CD4. Then that cells, they can be recognized specifically for HIV virus. But we all know that there is a lot of the virus can infect different species. So for example, the rabies, we call it a crazy dog, dog virus. And if the dog has not been vaccinated, and it's very easy to have this virus, and so far there is no treatment for that. Basically, if you get rabies, you're going to die because you're scared to the light, you're scared to the water, and finally it's become crazy and start to biting people because they invaded into the neurological system really quickly. And not only happen on the dogs, skunks, humans, and the bats, they're all going to happen. But we do have a plan. Some people said in 2030, the United States is going to be conquered the rabies. It will never be a problem for anybody. We'll see what that being achieved by that goals. This is the important, which tells you the steps of the viral multiplication. So how they're going to go to the host cell, and how they integrated the genome in there. So the steps including the first one is attachment, the second one is entry, then you uncoating of the genome, release it, then using the host cell material to biosynthesize their own needed nuclear material, and then assembly, and finally will be released. So we had a different section here. We talk about attachments. So the attachments have a difference between non-enveloped and enveloped virus. And the non-enveloped virus, we also call it a naked virus, because the only target they can do is a protein. So obviously, capsid is their attachment sites. Enveloped virus is a protein in the membrane, because the membrane, the envelope, they have composed by the protein. Then the attachment sites, they recognize receptor sites. Then there are multiple locations could be, like some membrane proteins, polysaccharides, and the lipids. We mentioned about that in the target cell. And when they do very interesting, like we talked about before. The process we call it a fuse, and then will generate is a receptor-mediated endocytosis. And very surprisingly, this endocytosis is kind of like we talk about AB toxin. It is a low pH. The pHs are usually about like 4 or 5. And finally, the viral particle is broken, and the nucleic acid is released. We call it uncoating. So this is very